Hello everyone, my name is Tianyi, a PhD student from Wageningen University and Research. Today, I will share my work on potato plant disease detection by integrating UAV-based hyperspectral and RGB imagery. Potato is a major crop worldwide, but potato grows face significant challenges due to high pest and pathogen pressure, which can lead to an estimated 32% yield loss. Monitoring disease occurrence is essential for timely interventions that mitigate economic losses and enhance potato quality. Among these threats, black leg disease and potato virus Y are particularly damaging. The symptoms of potato diseases could be captured by the sensors. In black leg disease, the infection spreads from the rooting seed tuber up to the stems, and it changes the structure and composition of canopy leaves. Similarly, in PVY infections, symptoms vary depending on the strain of PVY, but often manifest on the leaves and leaf veins. These visible changes provide a chance for sensor-based monitoring to detect the infected plants. Traditional disease monitoring methods rely on manual field inspections, which are labor-intensive, require specialized expertise. UAVs equipped with virus sensors provide an efficient and precise method for detecting diseases in potato crops. In this study, we collected the RGB and hyperspectral imagery at Lelystad in 2022. RGB was acquired with Demius P1, while hyperspectral imagery was collected with a line scan sensor, which has 269 bands, ranging from 400 to 1000 nanometers. A field inspector from the Dutch General Inspection Service for agricultural seeds and seed potatoes conducted a visual scouting, providing ground truth of the disease occurrence. A modified ULO-V5 network was employed for disease detection. To meet the input requirements, the RGB images were cropped into 640 by 640 pixel tiles with a 50% overlap. The hyperspectral imagery was spatially aligned and cropped accordingly to match the RGB tiles. The dataset was divided into training, validation, and test sets. Plant counts in each set as shown in the table. 7 different input configurations were introduced to the model for detecting infected plants, aiming to compare the effectiveness of monomodality and multimodality fusion for disease detection tasks. Vegetation indices were selected based on the top 15 most important features, which were determined by the coefficient weights of a linear kernel support vector machine model. The subbands indicate those bands used for vegetation index calculations. The evaluation results on the test set indicate that RGB imagery alone provides better detection accuracy than hyperspectral information alone. However, integrating hyperspectral information further improves detection performance. Notably, combining RGB imagery with vegetation indices yields both high precision and high recall, that demonstrating the benefits of feature selection in enhancing detection accuracy. The numbers in the PVY columns vary significantly due to the limited sample size, which greatly affects the evaluation matrix. For example, in the test set, there are only 31 PVY infected plants. If 21 plants are detected, the recall is 68%, but detecting 24 increased the record to 77%. This variability makes it challenging to conduct fair comparisons. Here are some visualization results. Blue boxes and yellow boxes indicate PVY infected and black leg infected plants respectively, while orange and red triangles represent false positives and false negatives. Here are the results from black leg infection subregion 1. These are the results from PVY infection subregion 4. So, we conclude that the selected feature, like vegetation indices, combined with RGB images, can improve disease detection accuracy. The PVY is harder to detect than black leg infection. That is because its symptoms appear on leaves, while black leg infection affect both the color and the plant structure. What's more, the class imbalance can cause the model to favor the majority class, means healthy ones. Additionally, advanced fusion techniques could further improve the detection performance. Thank you for your attention.